Good evening, ladies and gentlemen. It is my pleasure to present you the results of the research Prenatal Androgenization and Behavior and Insight from the Animal Model. From the conception through the fetal life, birth and puberty, brain development passes many critical points. In humans, by the end of gestational week 8, the rudimentary structures of the brain and central nervous system are established. Brain development continues during fetal period and for extended period postnatally. Despite the doubts of some scientists, there are true biological sex differences in the structure and function of the brain. Imaging techniques have revealed structural differences in various brain regions. For example, females have larger hippocampus and corpus callosum, while men have larger amygdala and ped nucleus stria terminalis. Development of the brain resides primarily in the genetic potential. However, environmental influences, especially hormonal milieu, can change expression of genes, activity on neuronal cells and circuits. The prenatal life is a vulnerable period of development, especially for the brain. Testosterone seems to be one of the factors that influences the brain via so-called organizational effects. That means it influences the building of the structure of the neuronal network, leading not only to sex differences, but also to individual variability of the brain morphology. This variability leads to differences in the brain function, including behavior, social interaction, and cognitive abilities. Studies have shown that prenatal testosterone concentrations are associated with increased risk for several different psychiatric diseases, including eating disorders, schizophrenia, and autism spectrum disorders. Androgen signaling is important in the development of sex differences in anxiety and aggressive behavior. Women with PCOS exhibit high circulating androgen levels even during pregnancy. Almost 40% of women with PCOS are diagnosed with at least one psychiatric disorder, such as anxiety, depression, or an eating disorder. Studies in humans and animals have shown that maternal hyperandrogenemia results in increased anxiety-like behavior in female offspring. Over the past several decades, anatomical and functional imaging studies in humans have highlighted several brain regions that are abnormal in patients suffering from anxiety disorders. The most prominent structures that are affected in patients with anxiety disorders are the hippocampus, the amygdala, medial prefrontal cortex, hypothalamus, and the ped nucleus of the stria terminalis. Hippocampus play a critical role in both cognition and mood. Reduced hippocampal volumes have been described in patients suffering from anxiety disorders and PTSD. The dual role of the hippocampus in both memory and mood-related processes can be explained by functional segregation of the hippocampus. In rodents, dorsal hippocampus is preferentially involved in spatial learning and memory, whereas ventral hippocampus plays a preferential role in stress response and anxiety. Besides pyramidal neurons, hippocampus contain also GABAergic inhibitory neurons, which are thought to play a key role in the local synchronization of and temporal coordination of pyramidal neurons. On the other hand, potential role of neurogenesis in affective states and anxiety is a matter of debate. Adult hippocampal neurogenesis is generation of new functional dentate gyrus cells through life and their integration into the existing neuronal circuits. Deficit of hippocampal neurogenesis increases anxiety-related behaviors and induces malfunctions of hippocampus-dependent processes and behavior. Brain-derived neurotrophic factor, or BDNF, exerts anti-apoptotic and pro-survival activity, modulates NMDA receptor-dependent synaptic plasticity, and enhances dendritic growth and branching. The precursor of the BDNF, so-called pro-BDNF, can determine neuronal fate by promoting death or survival of the neurons. Having in mind everything that was mentioned, the aim of this study was to examine the effect of late prenatal androgenization on reproductive phenotype and litter characteristics, serum concentration of testosterone, estradiol, progesterone, and corticosterone, the effect on anxiety-like behavior, GABAergic interneuron expression, and hippocampal neurogenesis by examining the expression of BDNF, Ki67, and NEO-N immunoreactivity in the hippocampus of the female offspring. Female Vista rats, 75 days old, were mated with the male rats. The first day of gestation was confirmed by vaginal smear. Pregnant dams were randomly divided into two groups, testosterone and control group. Testosterone group was treated with testosterone undecanoate subcutaneously on the 20th day of gestation, and the control group was treated with castor oil. Pups after birth, pups were housed with their mothers until 21st postnatal day. After birth and after weaning, the reproductive phenotype was determined by measuring anogenital distance, anogenital index, estrus cycle duration, and also characteristics of the litter were examined, like litter size, sex ratio, and litter survival. Between 70 and 85 days of age, behavioral testing was carried out during bright phase. After behavioral testing, brain tissue sampling was undertaken, and serum hippocampus and cortex was collected for further analysis. Further analysis including hormonal assays for measuring of serum testosterone, estrogen, progesterone, and corticosterone concentration. BDNF and pro-BDNF protein expression was determined by Western blot, and immunohistochemical analysis was employed in order to determine the expression of GABAergic interneurons and to examine the markers of proliferation Ki67. The elevated plasmase and the open field tests were used to assess anxiety-relating behavior in rats. The elevated plasmase is comprised of 2 open and 2 closed arms, and in this test, rats were allowed to explore the maze for 5 minutes. The behavior and movement was recorded by a camera and subsequently scored by a blinding experimenter, and the analyzed behavioral parameters were time spent in the open arms, number of open arms entry, and the number of head dippings. The activity in the open field test was tested immediately after elevated plasmase for 15 minutes in a photocell-equipped activity boxes. An automated system fully equipped with infrared sensor and its accompanying software was used to monitor rat behavior. The analyzed behavioral parameters were total ambulatory distance, time spent in the center, and time and number of entries into the center. The results were as follows. Prenatal testosterone administration decreased litter size at birth and litter size at weaning. It also altered sex ratio in favor of female offspring. So in the latter, there were significantly more female than male pups. Late prenatal androgenization did not induce masculinization of the female offspring because anogenital distance, anogenital index, estrocycle duration, and ovaries weight did not differ significantly among the groups. Concentrations of testosterone, estradiol, and progesterone were not affected by prenatal testosterone administration, but on the other hand, corticosterone concentration was significantly lower in testosterone group. In the elevated plasmates, prenatal testosterone administration significantly reduced time spent in the open Rs as compared to the control group. While in the open field test, the indicators of anxiety-like behavior such as time spent in the center and number of entries into the center were significantly reduced in T group as compared to the C group. These results indicate that prenatal testosterone increased anxiety-like behavior. Immunohistochemical staining with antibodies against NPY and parvalbumin allowed examination of the distribution of GABAergic interneurons in the hippocampal section CA1, CA2-3, and dentate gyrus in the offspring of both testosterone-treated and control female offspring. We can see here that prenatal testosterone treatment resulted in a significant decrease in the number of NPY immunoreactive neurons in the CA1 region of the hippocampus. The expression of parvalbumin in CA1 region was also significantly lower in the testosterone group compared to the control group. CA1 region of the hippocampus is important because CA1 pyramidal neurons carry the primary output of the hippocampus to other brain regions. Female offspring prenatally treated with testosterone had significantly increased mature BDNF protein expression both in hippocampus and the cortex. On the other hand, pro-BDNF protein expression was increased only in the hippocampus. Prenatal treatment with testosterone decreased the number of Ki67 positive cells in the hippocampus, which indicates decreased proliferative potential, that means decreased neurogenesis. Paradoxically, the number of neo-N positive cells in the dentate gyrus of the hippocampus was higher in testosterone-treated rats. Based on our results, we can conclude that late prenatal androgenization of female rats with testosterone undecanoate changes litter characteristics, decreases litter size, and changes sex ratio. Prenatal androgenization increases anxiety-like behavior, which is associated with decreased expression of parvalbumin and NPY positive cells in the CA1 region of the hippocampus. Late prenatal androgenization also decreases proliferative capacity, that means Ki67 expression in dentate gyrus of the hippocampus, which correlates with increased anxiety-like behavior. The expression of mature and mature BDNF and pro-BDNF was increased in the hippocampus, and this late prenatal androgenization decreased serum concentration of corticosterone without affecting serum levels of testosterone, estradiol, and progesterone.